I've got about 3 hours of questions here. What kind and range of money would be needed to run a national biobank with samples of all the endangered species?
It's such a damn good question, Kevin, and it's one that I don't have an answer for. But I know it'll take an act of Congress to get federal dollars to do it, and we should get that through congressional dollars. I am talking to people at the national offices about this, of what it would take. So I'll tell you, the federal storage facilities are there. The databasing infrastructure is more or less there. The challenge of getting field biologists to actually add this extra step in their process, it's not a big deal. They actually like doing it. They understand the value. And in many cases, it's a one-off. I mean, they may get one at a time. The cell culturing with mammals, not a big deal. So, probably a million going into new research on cell culturing is definitely a number that is going to have to happen. And that could be for birds as well as other species. But I think the biggest challenge is actually getting the mandate to do it. And my goal is to actually get it written into these recovery plans, which the biologists have to do as part of their work. It's, this is their job in the U.S. is to protect these endangered species. And this is our American legacy if we do it right.
You give the example of where you've worked with other organizations to help accelerate acceptance and to overcome things that just seemed, what are, in the political realm, what are things that need to be done? Maybe not by Revive and Restore, but you said conservation takes time.
And that's correct. What would speed it up? I'm not so sure that it needs to speed up in some ways. This is working at the pace of nature. There's plenty of bureaucracy that I'd like to speed up, and but I think that it's actually been a kind of a healthy thing. When Stuart and I started this, we wanted to socialize these ideas, to have public dialogue about it, and I think that's been good for the culture. People need to actually realize how important these technologies can be and to integrate them.
A very long, nowish answer. Two closely related questions here. And I'm going to start with Jenny, and then I'll ask the second one, which is anonymous. Which conservation work is Revive and Restore currently partnering with, or which would you seek to partner with in the future? And part two of that is, how do you find and choose your projects and partners? And how do you support them? What are the ways you support them?
So probably the biggest bang for our buck of all of those projects is just enabling genomic sequencing. Some of those projects we may fund $50,000 grants, some of them $250,000, a few up to $1 million over 3 years kind of thing. But it's really getting these foundational tools into their hands. And that's something that is just going to get cheaper to do, and ultimately you won't need Revive and Restore to do that, because they'll be able, as a conservation organization or land stewards, to be able to just make that happen on their own. But for now, we're kind of a gateway for a lot of these organizations at that front. The bigger projects, like the engineering, really thinking about how to advance reproduction for birds, those are much bigger projects. Now, back to the question of conservation organizations. When we think about how we're making a difference, our goal isn't to try these ideas, move the science forward. It's actually to integrate it into a functioning ecosystem. And we are never going to be in a position to do that. We need the nature conservancies of the world to take those genetically resilient sea stars, captive breed them, and reintroduce them to the wild. That's a huge process that has to happen by other organizations, not us. So we're the kind of bleeding edge, try it, de-risk ideas organization, enable people to start to use these tools, and hope that they get translated out to, the big NGOs.
So when you're starting a relationship You've been around for 10 years now. What does the arc of the beginning of the relationship, how do they typically happen, and how does it evolve, and when does it take off?
Well, we've done a really fun thing that we didn't know if it would work very well, but we called it, we started a program 3 years ago called Wild Genomes. And we asked for people to apply for research money to do sequencing on endangered or threatened species. But we put a very important requirement in it, that it had to be actionable, that it couldn't be just purely academic. It had to be something, information that once they gleaned from the genome, that they could apply in a way that it could actually help protect the species. And so, we've had way more proposals than we could ever fund. So we're looking for funding partners now as we build out that program. And so right now we have we just finished an open call around kelp ecosystems. And we did that in partnership with the Morris Animal Foundation and the Webb Family Foundation. And so they're kind of co-partners in it. And we actually have joint calls, and we have advisory boards that select the projects in a really good, healthy, functioning partnership. So I hope we can do more of that with funders.
Talk about unintended consequences.
Well, I have to talk about intended consequences because in the last 10 years, I always get the question of, we don't know about the unknown unknowns. And yeah, that's right, we don't. But we got to flip that question and say, what about the benefits of trying things? I mean, when I first started talking to coral biologists, and this was really early on, and we could see what was going on with global warming, almost everyone I talked to said, it is so desperate, Ryan. I mean, they were like going through PTSD diving. They said, it's so radical, we don't dare do anything to risk the unintended consequences of meddling with the reefs. And that has really flipped in the last 5 years. Mary Hagedorn and all the scientists that we're funding, they're actually saying we have to intervene. We may not have to genetically engineer. We may take those corals that are doing well on one part of a reef, and we may translate them to another part where they could still do better, or we could, help with breeding and repopulating. But some of them are actually open to where the technology will take them in order to save the reefs. So I think it's the risk-benefit equation that has to be played out, and it just depends on people's tolerance, and I've seen that tolerance shift.
What is a tool or technology that seems almost possible that would make your mission easier?
Stem cell technology. I have a really good answer for that one. So one of the things that I'm learning, and again, this is kind of drafting behind human medicine, is that stem cell technology is changing everything in human medicine. I mean, what did I just read the other day? That stem cells were being used. These are cells that are taken from your own body that are then implanted to reduce inflammation or to help with Parkinson's, all these different applications. But understanding how to develop those stem cells has been incredibly hard. Now, they've never been used for conservation, not really. And so because of that, Revive and Restore is going to host this fall a workshop. And I've invited now probably 20 different scientists that are doing stem cell research in all different fields, from veterinary medicine. Stem cells are big with horses. I'm an equestrian, and I can tell you many high-performance horses get stem cell injections. They do these things first with animals, and they do it with prized cattle and prized horses. And then they work it down to humans. I want to work it the other way and work it for wildlife. So we're going to gather these scientists together, and we're going to do a format that I've used in other workshops, which is really interesting. We've done it now, I think, 4 different times on different topics. And the secret to the success of this, and it really has been a success, is we get 30, 40 different people from very different disciplines to all work over 3 days, night and day, in a retreat facility. And we give them 3 different challenges that they all buy into. They elect and opt in to different work groups. And at the end of the 3 days, they're supposed to identify the problem, identify the solution, develop a roadmap on how to get there, and develop what they think could be the translational partners to make it happen, and the funders. And then I have a mock group of funders who were present on the 3rd day of the workshop. And I can tell you, the night before, these teams stay up half the night trying to get their decks together. And they pitch it as if for real. And what has happened on numerous of these workshops, the funders stepped up. And incredible research has moved forward. One in particular on gene drives for rodents on islands. It was incubated at one of our workshops. And so I want to do that with stem cells because no one's paying attention to it. And it's the secret ingredient that will allow all of these different projects to move forward for engineering.
There was a question that Kevin, I peeked at it, but he didn't give it to me. But I remember it was someone asking, could this technology be used to stop invasives? Like,
Oh, yeah.
could we do something with the Florida python?
Well, God, I really wanted to do something. It's a huge challenge reproductively with the python. So the technology that exists that's been worked on with rodents is called the gene drive. And what a gene drive does is it perpetuates very quickly a particular gene selection through reproduction. And when that happens, you could basically create sterility. Now, everybody is very concerned about a gene drive for one species, for example, getting rid of rodents on an island by introducing only males, right? You could just get that selection through, and all of a sudden you've decreased your population. But what if that rodent escapes off the island and then affects the good rodents other places? That's the big fear. So scientists are working on trying to figure out ways to create both self-limiting gene drives, and they have lots of different approaches to this, so that that unintended consequence doesn't happen, even should an engineered animal escape. But to me, it's one of the most humane things we can do, because what we do for these species, like killing thousands of Burmese pythons every year in Florida, if we could create sterility and remove them, it would be a much more humane thing to do. Using rodenticides on islands has had huge mortality on other species, especially raptors who eat those sick and dying and dead rodents, and they ingest the rodenticide. So, working with invasive species is huge. Now, where they're making progress with this, because I need to give you more success stories.
Yeah, we should, just a little.
This is a good one.
Oh, sorry.
It's a good success story.
Okay.
So Hawaii had mosquitoes introduced, I think it was in the 50s. I didn't study that stat, but in the 1950s, 1850s. Oh yeah, they came in, they definitely came in with the whalers, and they came in and shipped ballast and everything else. Those mosquitoes should never be in Hawaii. There are 5 species of in Hawaii, some that carry human pathogens, like chikungunya and yellow fever and all these other things. But there's another horrific one that carries avian malaria. That's what those honey creepers died of. And it is causing extinction like nothing else. The big challenge is, as these island temperatures rise, the birds who used to seek a refuge at higher altitudes where the mosquitoes don't go, now they can't go any higher, and the mosquitoes are going as high as they can. So there's no mountaintop to escape to. So people in Hawaii have known this problem, and they love their birds, and they've been completely conflicted about what to do with it. This is the animal-human conflict that just keeps playing out. And so gene drives were proposed. I was one of the ones who did workshops in Hawaii, and they just went, GMO, forget it. But there is a solution, and there are folks that came out of Google X Labs that have used what they are calling an organic substance called Wolbachia that they can engineer into the mosquitoes that causes infertility in the mosquito. And also, because of their incredible AI expertise, they've been able to grow these mosquitoes in great quantity because they can use AI to sex select, something that, we haven't talked about the role of AI in all this, but it's just going to be really transformative for things like captive breeding of small insects and everything else. And Wolbachia is now being set for trials in certain islands. It's really worked its way through from, I think, having the scariest idea of a gene drive to something that now is being called organic, being passable. But at the end of the day, it is engineered, and it will not absolutely knock out the population of mosquitoes. It will just drive them down, and they're going to have to keep reintroducing these mosquitoes over time.
Some years ago, I was absolutely startled when E.O. Wilson confessed that if all the mosquitoes disappeared from the world, he would not object. I mean, is this biological extremism?
Well, look, I was a big proponent. I did a workshop, and I called it mosquito-free Hawaii. I wanted to get them all.
Well, getting off Hawaii makes sense if they were invasive,
Yeah.
But off the entire planet?
No, but it's complicated. Yeah. There, many species of mosquito. They're not all bad.
It is not a charismatic species. So Adam asks a question. With so many extinctions and populations in decline, how would you prioritize which species you focus on?
Yeah, well, so as an entrepreneur, I'm purely opportunistic. I look for passionate people. I look for science that's going to be really applicable, that can make a difference. Those are the biggest things that we look for. At the end of the day, when we put all the different wild genomes proposals together, we try to look at, is there any synergy between teams that we could actually, if somebody's working on one part of the coral challenge and another, could we put them together? We look for things that are outliers. The project One People, One Reef, and I have two people from One People, One Reef right here. Nicole Crane and Giacomo, who told you about his genomic sequencing. We look for people that are really making a difference and that are bringing us sort of a broader view of this whole challenge of integrating technology. And I think Magul did such a nice job in that very short video that he did from a very remote island at, I think, some ungodly early hour to help me with my talk. Weighing that whole challenge that they have between traditional knowledge and new innovative science, and how do they understand more about what's going on with their fish that they depend on. Because, typically environmentalists come in and they say, oh, you just need to create a marine protected area. Just wall it off. And you can't do that when people are living on it. But can you say, well, let's have science help us tell you how healthy that population is and at what density it needs to be, or that coral reef, what challenge they're having with their livelihood. But I think that project in particular is profound in that it truly is collaborative. They are really learning from each other. And I think, that is really my goal, not on traditional knowledge, but I gotta say, conservationists are very traditional. I know you're using it in a different way, but it is an old school approach. This, wall off and protect is one school of thought. And they are completely anti-intervention. I mean, there were people when the California condor was going to go extinct that said, let them die with dignity.
Right.
I mean, David Brower, this dearly loved person, said, let them die with dignity.
They freaked out when they started using condor puppets to feed the condors. They said that would ruin condors forever, that they'd just become fast food condors.
And there are now 350 condors flying in the wild.
On that note, let's all give Ryan a hand. Thank you so much.
Thank you. Thank you, Paul.